J'introduis maintenant Guillaume Favre, qui va concourir hors catégorie, puisque les deux papiers ou les deux présentations qu'il va faire ont été publiés, excusez du peu, dans des journaux du Lancet, et avec un impact facteur qui vient de sortir pour ce nouveau journal du Lancet, qui a plus de 20, ce qui est assez impressionnant. Et ceci est le fruit non seulement du travail de Guillaume, mais de tous ceux qui ont collaboré au niveau suisse, et il y a eu beaucoup d'entre vous qui ont collaboré à ce type d'études, et je les remercie déjà. Guillaume, à vous.
So I will do the presentation in English for everyone. Thank you for the introduction. So we're going to speak about COVID-19, and we will focus on the vaccination, and especially the vaccination during the first trimester of the pregnancy. So we know that pregnant women are at higher risk of COVID-19 and severe COVID-19, and we know also that COVID-19 vaccine during pregnancy are reported to be safe and effective for severe disease. But very few studies assessed the early vaccine exposure, and this represents the organogenesis period, the higher risk of teratogenicity period. So the aim of our study was to assess the risk of congenital malformation after COVID-19 vaccine during the first trimester compared to pregnant women exposed in the second and third trimester. And we will see after why second and third trimester exposure. So we used the COVI-PREG registry based in France and Switzerland, and this was a multi-center prospective study conducted from March 2021 to March 2022, where we included pregnant patients who were exposed to at least one dose of mRNA COVID-19 vaccine. And the inclusion was made at the time of vaccine injection or just the visit before the vaccine injection. We defined the exposed group as any pregnant woman who were vaccinated from conception, 2 weeks after LMP, to 11 weeks of gestation and 6 days. And we didn't have a control group. So we decided to have a reference group composed of pregnant women vaccinated after 12 weeks of pregnancy, because it was representing the period none at higher risk of teratogenicity. So we didn't call it the control group, but a reference group. And our primary outcome was congenital malformation, and it was defined as one or more major and or minor malformation based on the EUROCAT classification. So we asked 2 experts on the field to classify which kind of congenital malformation was it based on this classification. And the secondary outcomes were live birth, stillbirth, and spontaneous abortion after vaccination. So we included 1,450 pregnant patients exposed to the COVID-19 and split into the first trimester exposure 124 patients and second, third trimester exposure 1,326 patients. And we see that we have only one congenital malformation in the first trimester exposure, representing 0.8% of malformation. And we had 11 congenital malformation in the second and third trimester exposure, which represent 0.8% also of malformation. If we test for association for crude analysis, univariate analysis, we have a risk ratio of 0.89 with the confidence interval including the 1. And if we adjust for a confounding factor, we have 1.01 of risk ratio, again with a confidence interval including 0. And if we see, if we look at the congenital malformation in the first trimester exposure, we had a syndactyly on the right hand of the fetus after an injection of the vaccine at 5 weeks of pregnancy. Regarding the secondary outcome, we had a rate of live births of 97.5% in the first exposure group, with 2 early spontaneous abortion and 1 late spontaneous abortion, which is similar to historical cohort. And in the second and third trimester exposure, we had 99.7% of live births, with 1 stillbirth at the end of the pregnancy, very delayed from the injection of vaccine, and 2 late spontaneous abortion, again a low rate of this outcome. So if we see our results, we can suggest that COVID-19 vaccine in the first trimester show no sign of increased risk of malformation compared to the second and third trimester exposure. But this study is very limited, and we will focus on three main limitations. First, we had a small sample size exposed in the first trimester, only 124 patients. That made impressive risk estimates, so it's very difficult to highlight small signal. We had low proportion of congenital anomaly. We had only 0.8% of congenital malformation, which is very low, and which can be explained maybe by the very short follow-up of time we have, because we followed neonates only 5 days after birth, which is very short, and we know that a big amount of malformation are diagnosed after the first week after birth. And we didn't have a control group, we have a reference group, and it's not an unexposed group. It was exposed during the second and third trimester, so it's kind of something comparable, but not a real control group. So if we need to have a message to go home with, is we had evidence for vaccine safety. We didn't show any big signal of malformation after the first trimester exposure, but the results are very limited, and there is a need of further study to confirm these results. Thank you very much.
Est-ce qu'il y a des questions? Is there any question? Yes, Daniel. I'm not sure it's working.
Yes. Thank you for this great work. Could you explain us how the transport works from the mRNA vaccine through the placenta? Is this an active transport to the embryo? And how is this? Is there an accumulation in the embryo, or do you know any studies about this?
I know this is a passive transport in the placenta, but I don't really know how it reacts after the passage through the placenta. And I don't know the reaction in the fetus, actually. I know that there is some studies analyzing the amount of antibodies we can have after birth and everything, but I don't really know the mechanism of everything.
Maybe I will answer. So there is, of course, an active transfer of the antibodies of the mother. The mRNA. But the mRNA do not transfer to the placenta.
Oh, yeah, sorry, yeah.
If you do PCR, RT-PCR on the product of consumption, if there is miscarriage or things like that, you will not find mRNA going through the baby.
So you don't find mRNA.
Yeah.
So how would be the mechanism for malformation if it does not cross the placenta?
Tricky questions. I think there is a lot of things we don't really know, and this is some of basic science to see if there's something we should look after, and actually we have no signal to be afraid of anything, so that's great, but yeah.
You have always to convince anti-vax, even if on the scientific point of view it seems evident, you have to show them. There is a court action against anti-vax for those who promoted the vaccine in Switzerland, Swiss Medic and us. So you have always to add data, even if on the scientific point of view it seems evident. We might imagine that some kind of inflammation is induced by the vaccine, as this inflammation might impair the placental function or whatever. But Guillaume shows that it seems not to be the case. Thank you, Guillaume. Oh, sorry.
One question. Why the choice of taking not a control group?
Because we didn't have any control patients. We didn't have it in the registry because the inclusion process is very time consuming. We need a few people to have it, and we concentrated on only vaccinated people, and we don't have in the registry people that are not vaccinated and no COVID.
So now we will listen to Dr. Radan.